Thank you for joining us for today's virtual session on intrinsically disordered proteins. As our host just said, my name is Robert Levenson. I'm located at UC Santa Barbara, where I'm a postdoc, and I'm excited to be participating in chairing this virtual spotlight session. And I'm looking forward to hopefully presenting, but certainly hearing some great scientists from our speakers. And so with that, I will jump into my first presentation. Okay. So I will be speaking today about charge neutralization, tuning the dynamic rest of the reflectin proteins. And I'm going to jump right into it then. So cephalopods, the squid, cuttlefish, octopus, are renowned for their beautiful dynamic coloration, which they use for camouflage and communication with each other. While one form of coloration available is iridescence, and that gets produced by iridocyte cells. And as you can see here on the figure, this is found in all types of cephalopods, but notably in loligynid squids like Doritus's opalescens on the bottom left here, the common marked squid. This iridescence is tunable. These cells can be activated. And so if we look at a confocal microscope of fixed iridocyte cells here, these iridocyte cells are filled with reflectin proteins, and that's the green, in close proximity with membrane surfaces. If we look by electron microscopy, you can see a lamellar structure. If we zoom in, we can really see this lamellar structure. And what we're seeing here is the dense areas, the dark areas, are the intracellular reflectin interspersed with extracellular medium. There's a difference in the refractive index between these two areas. And so as light travels through this Bragg reflector, at the interfaces, light gets reflected, and based upon the spacing and refractive index contrast, we generate light of, we reflect light of different wavelengths. Now, this iridescence is regulated in loligynid squids, and this is done through an acetylcholine based pathway. Acetylcholine binds to a muscarinic acetylcholine receptor, activating a kinase that phosphorylates these reflectins, driving condensation and the emergence of tunable iridescence. Importantly, this is accompanied with water expulsion from the cell that further increases the density of these reflectin lamellae and changes the spacing. Now, the reflectin proteins themselves are overall cationic. They are intrinsically disordered, of course, and they are block copolymer-like proteins. So they have multiple tandem repeats, and those are composed of highly conserved domains. These are the RMs located here with this unique and and highly conserved patterning, as we see on the bottom, and then lesser conserved linker regions, the narrower lines there, which are weakly cationic. Those linkers are the regions of phosphorylations in vivo. And in this work, I'm going to focus on our work with reflectin A1 here on the top. It's been a real workhorse for understanding. So we can express reflectin A1 in bacteria, purify it, and purify it in acidic conditions, solubilize it in acidic conditions. And then we can drive its assembly as a surrogate for the in vivo phosphorylation process. We can drive its assembly by pH neutralization, so diluting it in the buffers of higher pH. And if we do that into incrementally high pHs labeled here on the figure, we get incrementally large spheres of tunable size and low polydispersity. The low polydispersity of these assemblies is confirmed by X-ray scattering, good agreement between X-ray scattering and the DLS results that are shown here, as well as TEM that shows the monodispersity of these spheres, as well as their spherical nature. Further characterization of this assembly process by circular dichroism shows us that in the monomeric acidic state, the protein is disordered, that strong negative peak at 200 nanometers. And then upon assembly by pH neutralization, we see a change in the CD spectrum, suggesting conformational changes in the protein. electron paramagnetic resonance shows that in the acidic monomeric state, the protein is or the label is highly dynamic, whereas upon assembly through pH neutralization, dynamics are greatly decreased. The spectra is highly broadened. And so this is consistent with reflectin assembly progressing the final assembly being dynamically arrested. So there's very little dynamics of the monomers that have assembled together to form these larger spheres. This is also confirmed by a simple DLS assay where we add aliquots of monomeric protein into a buffer that drives assembly. And as we add increasing aliquots of monomer, each set of monomer assembles separately to form particles of the same size. And so the overall size does not change. There's no kind of dynamic exchange going on. And the amount of light scattering signal increases linearly with aliquots. To further understand this process, we generated a series of mutants. Our first was a set of mutants that made phosphomimetic glutamate mutations in the sites previously located to be phosphorylated in vivo. And we add increasing numbers of negative charges, increasingly neutralizing the protein. Another series of mutants removed various segments of the protein. And we performed that pH neutralization assay on all these mutants. And what we found is that the assembly behavior of all these various mutants and wild type could be predicted based on a calculated net charge of that particular protein at a certain pH. And so this diagram summarizes the assembly behavior of all the mutants that we saw before. In conditions that have high net charge, so acidic conditions, the protein remains monomeric due to electrostatic repulsion between monomers. And then upon neutralization across a threshold, a charge threshold, we get the formation of assemblies of tunable, predictable size based upon the calculated net charge. We also produced a mutant that removed those highly conserved domains. So this just consisted of the linker regions and purified and characterized this protein. And it assembles in the basic DLS pH neutralization assay. Similarly to wild type, it has a similar charge threshold. So those highly conserved domains are not at least required for the basic assembly process that we're seeing here. And further characterization of the assembly of this LO linker-only protein shows that indeed it's forming relatively monodispersed assemblies, a little more polydispersed in wild type, but similar. And it undergoes similar conformational changes upon assembly at pH neutralization. Electron microscopy or atomic force microscopy characterization of these assemblies shows that they are spherical, particularly at the large size area. It's highly spherical. Clusters of particles can be sometimes observed, and they undergo deformation. It's soft matter that we are assembling here. And the very high degree of sphericity that we see in these assemblies strongly suggested to us that there's some kind of a liquid state, either transient or not, involved in the assembly process, followed by dynamic arrest to give us the size stability that we see in our tests. So these pH neutralization assays I've been speaking here were all done in very low ionic strength, non-physiological conditions. And so we wanted to see what are the effects of salt upon this process. And so here we've got a constructed phase diagram of A1 assembly as a function of both pH on the x-axis and salt on the y-axis. And we see at lower pH, a greater degree of salt is necessary to drive assembly, consistent with a greater degree of screening required to drive that assembly. At pH 5, if we dilute our monomeric protein with no salt in the buffers, we'll have increasingly more salt. And we characterize those, we get spheres that are progressively larger with more salt. In this case, these assemblies are size stable over time. They do not continue to grow as we add salt. And so analogous to the pH neutralization assay where we increasingly neutralize the protein and drew larger assemblies here. And again, they are spherical as well. So similar morphologies to that seen by the pH neutralization assay. If we increase the salt even further, we go up to 250 millimolar or beyond, we obtain a micrometer, roughly micrometer size droplets that have liquid properties. Here we're working with a fluorescently labeled A1 protein with fluorescein attached to it for observation by confocal microscopy. And characterization of the liquid properties of these assemblies shows that they have pH-dependent dynamics. So at low pH, pH 4, there is a recovery, fluorescence recovery after photobleaching. And however, in contrast to that, at higher pH, at pH 8, the amount of recovery is very, very small to no recovery at pH 8. So at this nearer to physiological pH conditions, the dynamics are greatly reduced. These particles are quite solid. And consistent with that, if we look at, say, droplet fusion at this high pH, we never get complete droplet fusion. Instead, we result after a long time in these kind of ellipsoidal shapes just because they're so solidified. To further kind of demonstrate that, here we've taken A1 reflectin A1 protein, applied 2 different labels, either fluorescein or rhodamine. If we take these 2 separate monomer proteins, we combine them together and then drive assembly by dilution into high salt. We get micrometer-sized droplets where homogeneously labeled with both labels. In contrast with that, if we take each independent label, we drive assembly in separate tubes and then combine after a very brief incubation period. In this case, it's 2 minutes with this example. The labels are now heterogeneously distributed. So some degree of fusion does occur, but the mixing within these droplets is so low that the labels do not become anywhere close to homogeneously distributed because they become solidified so rapidly. So what kind of forces are driving this LLPS and rapid gelation of the reflectants? It's been well established that there are a number of so-called sticker interactions that can drive liquid-liquid phase separation. So things like pi-pi interactions, cation-pi interactions, notably arginine cations and tyrosine aromatic rings. And charge-charge interactions, all these act as stickers to drive phase separation. If we just look at the compositions and distribution, the spatial distributions of some of these stickers with reflectin A1 on the top row here, compared with a couple other representative phase separating proteins that are well known, reflectin A1 has a very high, the highest that I've seen in my inspections, contents of arginines and tyrosines forming those sticker interactions, which is known to contribute, a high concentration of these is known to contribute towards a very strong drive towards phase separation. Additionally, one note is that they're very low in glycine as well, which may contribute to the rapid gelation, one factor contributing to the rapid gelation. If we look at how these stickers are distributed across this block copolymeric-like reflectin proteins, they're very uniform distributions of the stickers, another attribute which has been shown to be associated with a strong drive towards phase separation. So these reflectin sequences seem to be strongly optimized towards phase separation. So to summarize, the evidence indicates that reflectin proteins drive a tunable and reversible optically active biocondensate system where acetylcholine drives phosphorylation, which drives phase separation of these reflectin proteins, forming a dense phase, excluding water. And that dense phase itself produces the refractive index contrast in comparison to the extracellular medium to produce iridescent light of tunable color dependent upon the parameters. And with that, I'd like to thank you for your attention, and my email's there if anyone has additional questions, and thank those who I've worked with both in the lab, in Dan Morse's lab, and outside the lab. And I'd love to take any questions you may have.
All right, thank you, Robert. We have some questions in the Q&A.
Great.
From Marina, did you try other salts other than NaCl in your assembly assay? I am thinking about possible effects of anions from the Hofstadter series. Thanks.
Yeah, so we've tried different cations, and so, for example, different cations don't seem to have a strong effect on phase separation. On assembly in general. So like calcium chloride versus sodium chloride, it all comes down to the chloride concentration. So it's the anion that seems to be the operative agent here. in terms of different anions, what we've seen is there's definitely a strong effect of the anion selection. So for example, phosphate anions very strongly drives assembly at very low concentrations of phosphate. So generally we need to stay away from that. But there is a very strong anion effect, absolutely.
Great. So from Randall, he wants to know, given that the droplets are kinetically arrested and this is responsible for their uniform size, how does size increase with increasing pH instead of getting more but smaller droplets?
So the way we see it is that upon pH neutralization, all reflectin assembles, and something about the charge distribution on these change is affecting the kinetics of both the growth and the arrest process, resulting in greater sized droplets that are kinetically arrested later in the growth process. When there's more neutralization compared to less neutralization. But the exact details of the effects are something that we're still trying to piece out.
Great. Jeremy wants to know, do all iridocytes in a given cephalopod express the same reflectin isoform, or is there diversity on the isoform expressed depending on the anatomical location of the iridocyte?
There absolutely is. Great question. In particularly in Dorichthys opalescens, the market squid I talked about, some iridocytes are tunable, whereas others are not. And indeed, there is a correlation of tunability with the presence of certain reflectin chains. There's a paper that came out of our lab maybe about 8 years ago that highlights the differences in the different forms of the reflectins. And it turns out there's another reflectin not discussed here called reflectin B, which seems to be particularly associated with the rise of tunability in particular. And so it's also, there's effects of mixtures as well in enabling that, enabling the tunability of assembly.
Awesome. I think we are coming up on time, but thank you so much, Robert, for a great presentation. And Robert will be answering the rest of these questions in the Q&A box.
Thank you all very much again. Okay. All right, so with that, it is my pleasure to turn us over to the next speaker we will have today, and that will be Jay-Z Wu at the Stowers Institute for Medical Research, located in Kansas City, Missouri, and he'll be speaking to us about the determinants of the varying modes of self-assembly of the protein TDP-43. With that, please go ahead and take it on.
Thank you. Okay, I'll share my screen. So good afternoon, everyone. My name is Jianzhen Wu. I am a senior graduate student from Hoffman Lab at Stowers Institute. Today, I'd like to share our recent work on dissecting the aggregation pathway of TDP-43 low complex domain inside cells. So proteins contain various physical states. The initially diffused protein monomer can condense into a liquid droplet, which can continue to solidify into a gel or transit into a solid or amyloid-like aggregates. In general, transition from one state to another will significantly affect its cellular function. In past several decades, the research focus was primarily on the amyloid, as amyloid aggregates are heavily implicated in the neurodegenerative diseases. However, accumulating data has suggested the important role of non-amyloid or pre-amyloid assemblies. Thus, studying the entire aggregation pathway, not the amyloid itself, is more crucial to understand their roles in both functions and disease. However, studying the full spectrum of protein aggregation is challenging, as I show you next. So commonly, to study protein aggregation, purified protein is put in the test tube, and the aggregation is monitored as a function of time. In theory, the pre-amyloid species or non-amyloid species, such as oligomers, forms first and then followed by the amyloid formation. Ideally, we would expect to see a mixture of pre-amyloid species and amyloid so that we can study their interrelationships. However, in reality, since the amyloid is known for its ability to propagate its structure, anywhere in the solution, once a single amyloid structure forms, it will then rapidly polymerize the available monomers in the entire pool and convert them into amyloids. In results, the amyloid species will dominate over the non-amyloid species, precluding the analysis of full picture of the protein aggregation. So how could we overcome this challenge and capture pre-amyloid species or non-amyloid species? One straightforward way is to isolate the amyloids and keep it from spreading in the other region of the test tube. We can achieve this by dividing the entire test tube into smaller compartments as shown here and create independent reactions. By analyzing the protein assemblies in all the compartments, we will get a better picture of the protein aggregation pathway. So in principle, the smaller the volume is, the better the resolution will be. With that, we take advantage of the cells to serve as better reaction compartments, given that the volume of a yeast cell is at the scale of femtoliter. Now we can capture the pre-amyloid species, but when we look protein aggregation in such a small volume at single cell level, the chance of capturing an amyloid event is extremely low, since amyloid nucleation event is very rare in cell size volume, which may partially explain why the pathological aggregates, amyloid aggregates, does not show up until a patient gets old. So to increase the chance of capturing the amyloid during the time scale of a cell culture, we can collect a large number of cells. This is feasible with the help of flow cytometer, or we can just wait until the amyloid aggregates show up. This is not feasible because we don't know how long we need to wait. It could be days, months, or even years. An alternative way is to increase the rate of nucleation, amyloid nucleation, by overexpressing the protein and provide an amyloid seed, which I will talk later in my presentation. With all these considerations, we developed a FRET-based flow cytometry approach called DAM-FRET to quantify the extent of a protein self-assembling at different concentrations in cells. Yes, as you can see, FRET is commonly used to report protein-protein interactions, and we use FRET to approximate protein assembly inside the cell. In addition, we use a photoconvertible fluorescent protein MUS 3.1 that can serve both as the donor and acceptor. Thus, a single construct is sufficient to report FRET, which reduces the noise compared to the traditional two-construct FRET. With this assay, we were able to identify these different phases for yeast prion protein subcertified with low complexity domain. To further push the boundary of understanding the protein's phase behaviors, we chose a well-documented protein, TDP43, which is an RNA binding protein and is involved in the RNA metabolisms. TDP43 can also form pathologic aggregates and are involved in 97% of the ALS. Today's talk will focus on the low complexity domain as shown here. The aggregation of TDP43 has been shown to be largely mediated by this low complex domain, or LCD, and most familial disease-associated mutations occur within this region. Though this protein has been extensively studied, a comprehensive picture of the TDP43 LCD aggregation pathway in a cellular context is still lacking. More importantly, since several labs has resolved the structure of some parts of the LCD, and this will allow us to interpret our data more confidently. Here is a density plot showing the distribution of cells expressing the LCD that is tagged with the MEIS. Each dot represents one cell, the individual cell, as I mentioned previously. The axis represents the protein expression as measured by the fluorescent intensity. The y-axis is the ratiometric FRET level, or AMFRET, which indicates the degree of protein assembly in each cell. From this plot, we can appreciate two parts of this distribution. The population that has negligible amphoret indicates the absence of protein assembly, which represents the monomer population. As the concentration continues to increase, the cell acquire a concentration-dependent increase in amphoret, indicating the presence of protein assembly. We figured out that the threat comes from the protein oligomerization within this population. Thus, we designate this population as oligomerization population. The kink position between the two populations reflects the concentration required for the oligomerization to occur. As you can see, in the oligomerization population, the protein shows diffuse pattern, even using the super resolution microscopy. I'd like to point out that the oligomerization population is difficult to distinguish from monomer population because they are all diffused if we just look at the cell under microscope. However, with our assay, by comparing the thread level, we can distinguish oligomerization from monomer. So other groups have identified an alpha helix region within this LCD and showed that this alpha helix mediates the cooperative oligomerization in vitro. To simplify, I'll refer the blue helix as anterior alpha helix and the red helix as the posterior alpha helix. So next, we are interested to see whether the helix mutations will affect the oligomerization in cells. First, we examined the helix enhancing mutation G335I. As you can see, now the oligomerization can occur at a lower concentration. This is consistent with the in vitro data from Foz's group, showing that enhancing the alpha helix increases the effective affinity between molecules. In contrast, when we use the helix disrupting mutations, A326P, as shown here, now the oligomerization occurs only at a higher concentration compared to the wild type, consistent with the role of helix in promoting the oligomerization. Interesting, this mutation does not completely wipe out the oligomerization population. Since S326P only disrupts the anterior alpha helix, we reasoned that if we continue to disrupt posterior alpha helix, we should eliminate this population. As expected, when we disrupt both helix segments with an additional mutation M337P, the oligomerization population is gone. In the meanwhile, a different population with lower thread level shows up. After checking the cells with this population, we found that the protein forms liquid condensates. From this, we conclude that in cells, the oligomerization is mediated by the alpha helix, and the oligomerization competes with the formation of liquid condensate. Next, we continue to explore the comprehensive picture of its aggregation pathway, especially the amyloid. To increase the chance of capturing the amyloid states, we take advantage of the presence of an endogenous amyloid seed called PIN+ in isogenic yeast strain, which can promote amyloid formation of LCD. When we express LCD in PIN+, we notice that it forms a high thread population and circled by this yellow circle. We also observe the beautiful fiber structures in this population, indicating the formation of an ordered structure. We refer this population as atypical amyloid population. So in alpha helix disrupting mutant, where we see a population with an even higher amphoret, and show in the blue circle indicating a more ordered structure. Using SDDH, we found that the atypical amyloid is sensitive to SDS, while the blue circle population form SDS-resistant smears, which is one of the characteristics of typical amyloid. Thus, we refer this population as typical amyloid. We further confirm that the typical amyloid is AMY-tracker positive, which is a commercially available amyloid dye, while the atypical amyloid is negative for the amyloid staining. When we compare the amyloid formation propensity among these alpha helix mutants, we find that the helix-mediated oligomerization competes with typical amyloid nucleation. As you can see here, the alpha helix enhancing mutant G335I completely block amyloid formation. As the helix content decreases, the population shifts towards to the typical amyloid. So in summary, due to the technical challenges, current research focus on each individual assembly in isolation. With STEM-FRED, we were not only able to capture both non-amyloid and amyloid species, and show here, we are able to capture 5 different states of TDT43 LCD in cells, but also we were able to dissect their interrelationships, which will help us to build a better picture of the aggregation pathway. I'd like to express my sincere gratitude to my mentor, Randall, who guides me and gives me full support on this project. I'd also like to thank my lab members, co-facilities. We are collaborating with Alex, Whole House, and WashU to explore the conformational changes within and outside of alpha helix with all atom simulation. And I also thank for the funding sources. With that, I'd like to take any questions. Thank you.
Okay, I'd like to thank Jay-Z for that wonderful talk. We have a couple questions in the Q&A. If you have any additional questions for Jay-Z, please enter them in there. But I will read the one of the first questions we got from Marina. Do you have more than one FRET event in your flow cytometry experiments, or are you only measuring dimers? One FRET event.
Okay, so basically the way we do these experiments, we put the proteins in the cell and we use the plasmid and express these proteins at very different concentrations. So each cell contains different concentration. By collecting all the samples, we basically can get all the population with individual cells. So the short answer is yes, we can capture all events which are captured in each individual cells.
Great. We have a question from, all right. We have a question from Radha Krishnan. As the low complexity domain is an IDR, how was the presence of 2 helixes identified and validated?
Okay, so yeah, TDP3D in this regard is special. So this is protein's intrinsically disorder, but that alpha helix segment is well conserved throughout the vertebrates, and it has been identified through the simulation first, and then they confirmed for this group, and 2 other groups also confirmed in vitro with NMR the definite form of helix in vitro.
Great. And from Gorak, how do you describe the importance of polar amino residues in oligomerization, nucleation, and amyloid formation?
So at this point, so since I just focus on the alpha helix region, which is only part of this domain, maybe only 1/4 of the entire domain. So within this region, there is hydrophobic patches. There's no like a strong polarity here, but I have data show outside those regions where have the polar amino acids, and we do see an effect which I don't have time to share with you today, but I'd be happy to follow up on these questions later.
So thank you again, Jay-Z, for that wonderful talk. And we will move on to our 3rd speaker of the session today, Eden Gallegos from Louisiana State University, who will be speaking to us about probing the dynamics of human 5-lipooxygenase. And take it away.
Thank you. Okay. Yes, I am Eden Gallegos. This May, I graduated from LSU with my bachelor's of science, and I studied biochemistry there and research inflammation. I'll be starting my PhD, my MD PhD this August in New Orleans, and today I am presenting my research on elucidating the open and active form of human 5-lipoxygenase. Let's see. First, a lipoxygenase, or LOX, is an enzyme that causes inflammation by oxygenating lipids to produce potent inflammatory molecules. Human 5-LOX resides in leukocytes, and when it's signaled to initiate inflammation, it moves to the nuclear membrane. Here, a phospholipase, PLA2, cleaves arachidonic acid, which is a lipid. 5-LOX then uses arachidonic acid as substrate in a two-step reaction. The first product comes from a dioxygenation reaction that adds molecular oxygen to arachidonic acid to produce 5-HP. Next, 5-LOX forms an epoxide, producing leukotriene A4. LTA4 can be converted to other leukotrienes and cause an inflammatory response itself. When the system is overactive, it can play a developmental role in diseases like coronary artery disease or asthma. So this is one reason we're interested in the structure and function of 5-LOX. So 5-LOX is a peripheral membrane protein, meaning it doesn't always stay or act in aqueous solution. It's also intrinsically unstable, but because of its importance, our lab made a pseudo wild type version of 5-LOX, referred to here as stable 5-LOX. This is a soluble, stable version of the enzyme that just allows us to study it in vitro. This model yielded a crystal structure for human 5-LOX, and here we see the active site iron surrounded by a mostly alpha helical region. It's not emphasized here, but the structure showed a certain difference compared to other LOXes that stuck out to us and gave us a hypothesis about the potential for conformational flexibility of 5-LOX. So helix alpha 2 is very different in the 5-LOX structure compared to other LOXes. While 5-LOX shows a broken V-shaped helix, other LOXes, such as 15-LOX2, show an elongated version. However, recent crystallization experiments actually revealed this elongated structure in 5-LOX. This is pictured to the right. The global structure is the published structure for stable 5-LOX with helix alpha 2 emphasized in pink. And then in the box, you see the broken helix overlaid with a gold helix with an alternate conformation for these residues. This alternate structure gave us the idea that 5-LOX could exist in an active state and a resting state. And if it had access to the elongated helix when in an active state, this would open a path for the 20-carbon substrate arachidonic acid to enter the active site. So all of this together and more led us to think that a conformational change might form this alternate helix. So next I'll explain how this could be functionally significant. Here's a movie showing what this conformational change might look like. Notice more space is created by bulky residues moving away from the active site. These residues are hydrophobic, and in solution, their exposure on the surface will be unfavorable because of the hydrophilic environment. And now we know that 5-LOX is a peripheral membrane protein, so this movement of residues made us wonder if a conformational change could also assist in membrane binding. Here's just a different angle of these structures down here. Notice how 2 phenylalanines are pointing downward in the long helix. If this is where the membrane 5-LOX interface is, then these Phs could be involved in something known as Phi insertion that is common for peripheral membrane proteins. It would make sense for the Phs that are favorably buried within the protein while in the cytosol to move outward when given the chance to favorably interact with the membrane. 5-LOX also associates with a membrane-embedded helper called 5-LOX activating protein, or FLAP. So the conformational change could have a role in that interaction as well. And then lastly, because 5-LOX makes strong inflammatory molecules, it would make sense if a structural change was also involved in catalytic regulation, because by creating that wider pathway into the active site, this elongated helix might allow for a faster reaction. So to test this hypothesis that helix alpha 2 changes form, we altered specific residues on the helix and then evaluated the impact on the global properties of the enzyme, as well as any alterations to the catalytic activity. Just in addition to knowing if an alternate form of the helix is available, we investigated these exact molecular determinants here just to give us a solid background for research that can treat disease states like structural-based drug design. For simplification, an open conformation refers to the state of 5-lox with an elongated helix, and closed means that 5-lox has that broken helix. So we design mutants to favor either the open or closed state. In green, you see 2 residues that we mutated to the neutral residue alanine to favor an open conformation. The glycine here is a known helix breaker, so we didn't want that present. And then the aspartate here forms a strong hydrogen bond in the broken helix, so we got rid of it too. In blue, you see 2 residues that we mutated to reinforce the favorability of a closed conformation. The Phes are already happily exhibiting hydrophobic interactions and also actually cation pi interactions within the protein. So we capitalized on nearby positive arginine residues and added negative aspartates to generate ionic locks. We first noticed some oligomeric trends when purifying the variants by size exclusion chromatography. Stable 5-lox could form monomers and dimers in a basic buffer, and then it was fully converted to a monomer when the disulfide bond reducing agent TSEP and chelating agent EDTA were added. The closed mutant was a monomer regardless of the buffer, while the open mutant was a dimer regardless. This first made us think, okay, if hydrophobic residues are exposed more often on that open mutant, such as what we see here circled in red, then what if hydrophobic interactions occurred between this interface of helix alpha 2 on the monomers? Are they looking for a binding partner within the aqueous solution? This also makes sense for the closed model to not need a binding partner if the hydrophobics are rarely exposed. And then if these hypotheses are true, this could also be important down the line for understanding membrane and flap associations also, right here as you see pictured on the bottom right. So just our next global evaluation was limited proteolysis, which is a technique where your protein of interest is incubated with a protease. And for us, that was pepsin. Pepsin cleaves at aromatic side chains. Oh, and I just want to point out that based on the size of polypeptide fragments produced, we know that we did not remove a protease cleavage site by mutagenesis. When this process is complete, we observed a relative change in SDS band size by comparing before and after proteolysis samples. So at the top, you see images of the raw data showing samples before and after 15 minutes of proteolysis. And then the bar graph depiction of this data is from triplicate measurements. We first noticed that stable 5-lox resisted proteolysis pretty well. There was not much of a change in band size. We also observed that the closed mutant resisted cleavage also, but the open mutant showed a significant increase in susceptibility, as only about 50% of that full-length protein band is remaining after 15 minutes. So the data supports increased polypeptide flexibility of the open mutant, and we interpret this to mean that the open mutant has some new properties compared to stable 5-lox, while the closed mutant mimics stable 5-lox activity here. With that, we confidently moved on into a closer look at that active site. So we tested the activity by monitoring the production of the first product of 5-lox 2-step reaction. That product absorbs at 235 nanometers, allowing us to observe an increase in absorbance over time. With the same concentration of enzyme and substrate for each variant shown in this graph, and no enzyme activators present, we saw 2 interesting results. First, the pre-steady state lag phase, which is, for instance, the wind up before the constant or steady state activity occurs, that was altered. The open mutant had virtually no lag phase, while the closed mutant closely mimicked the lag phase of stable 5-lox. Second, concerning the linear steady state activity, the open mutant exhibited a sharper slope, which coincides with a larger initial velocity of Hd production. Our interpretation of this data supports increased access to the catalytic site for the open mutant, which is consistent with our open 5-LOX model. And then further, the previous observations at one substrate concentration was actually reflected across many concentrations. Shown here are just saturation curves for these variants. Each dot reflects the initial velocity as specific arachidonic acid concentration, and larger initial velocities for the open mutant, and conserved initial velocities for the closed mutant compared to stable 5-LOX were the result. The K-cat, or product turnover number, was increased by a factor of 4 for the open mutant. And then similarly, the substrate affinity, reflected by the K-half here, was around 4 times greater for the open mutant, as higher affinity is associated with a lower K-half. Lastly, the data fit to sigmoidal curves, which is consistent with the idea of a conformational change associated with enzyme activity, considering that this activity held true for both monomers and dimers. Across the board, the open mutant appears to be acting in a way that suggests increased access of the substrate to the active site, or increased product release, or both. The big picture here is that 5-LOX inflammation as a whole has a big role in many diseases we face. And if we can understand that underlying function of 5-LOX structures, we're close to understanding how disease states come about and how to design treatments that will be effective against an overactive inflammatory response. So I just want to say thank you to my mentors, Dr. Marsha Newcomer and Nathan Gilbert, for their guidance throughout both my experiments and projects and undergraduate experience. And thank you also to my entire team for your support and collaboration. Thank you for watching my presentation. I hope you enjoyed it, and I'm happy to begin answering questions. And my email is below if you have further questions.
Great. Thank you, Eden, for your talk. If anyone else has additional questions for Eden, please submit them through the Q&A box. We have a couple questions here from May. We have a question: which factors favor the enzyme to be in active state in vivo?
Well, so we do not actually study them in vivo. So this is we are proposing something new that has not been discussed before. So I'm hesitant to comment on that, but we would think our idea would be that in the cytosol, it would be in a resting state, in a closed state, so that you don't have spurious activation and lead to inflammatory molecules just acting, out of place. And then when it is signaled to move actually by calcium to the membrane, then it will form that open helix when it interacts with the membrane and that helper protein and things like that. So that's our idea.
And do you know where the substrate portal is for 5-LOX? Is it located in the helix that opens and closes?
Yes, well, the substrate access portal, I think, is around there. There's another helix that I'm forgetting the name of right now that also plays a big role in that. But yes, it surrounds that substrate portal.
There's a question from Song. Do you have ATP present in your experiment? If not, what effect do you anticipate if you add ATP?
So we didn't want any activators present that have been used previously. So we did not have ATP because we wanted to solely see impacts of the mutagenesis. And so yeah, we did not have it. And if it was there, I think if any activators were there, we would see some increased activity in general. But I do think since we have tried a few activators actually, COVID kind of cut it off a little bit, but we did see the same results reflected as when there were no activators.
Okay. And we have one last question from Monica. What changes, the question is, what changes in amino acids render 5-LOX stable? Not sure.
So that actually was work done by Nathan Gilbert here and Marcia Newcomer. And actually, I would ask that if you would like follow up on that question, they would be able to answer that, and you can email me and I can send that over to them. But they designed that back when he was doing his PhD.
Great, great. And there's a question from Peter saying, do the regulatory phosphorylations influence the conformations of 5-lox?
That is a good question. We have not yet gotten to that point, but I'm not quite sure, actually. That's something I would like to know the answer to as well.
Well, if there are any further questions, please feel free to contact Eden through the email there. With that completes my presentation and our presentations today. And I will thank you all for attending. Thank you very much to the speakers.